Good morning, everyone. Welcome to today's grand round. It's a great pleasure to introduce Dr. Shihuan Kang, who's a professor and cancer center chair in stem cell at Purdue University. Dr. Kang is a really very well-known scholar for his pioneer work in the stem cell research. His lab studies the development of and regeneration of skeletal muscle, as well as cells such as adipocytes that interacts with muscle stem cells. He has over 200 publications, over 13,000 citations, and he has served as a member of over 50 grant review panels and won numerous awards. So today he's going to tell us how lipid droplet dynamics regulate muscle stem cell fate. So without further ado, I will pass the microphone to Dr. Kang.
Thank you so much, Dr. Chen, for the invitation. It was a really great pleasure to talk to scientists and clinicians at the University of Minnesota. It was actually funny. I came to the medical school at the University of Minnesota last month and did a seminar for another group, physiology group. So it's really exciting to see the campus, the breadth of research on campus there. So what I will try to talk today is about the story we published recently, but we're still working on this. That's why part of this is unpublished. So does everyone hear me okay? I just want to make sure.
Yes.
Okay.
Yes.
So yeah. So what we are looking at here is a section of a muscle, a skeletal muscle. All the green cells here are muscle cell, or we call them myofibers. So this muscle is a little bit unique in that it is during regeneration. So what we can tell is that the regenerative muscle from a non-regenerative muscle is that during regeneration, the cell nucleus actually are in the middle of the cells, but in a nice regenerated muscle, the nucleus are usually in the peripheral. What I want to emphasize in this section is that we also see those red cells here. Red is marked by a dye called oil red, which marks lipids. Okay, so suggesting that in a regenerating muscle, there's a lot of fat cells also play a role in this regeneration process. So the regeneration itself is mediated by a cell pointed in arrow here. These cells are in the peripheral of a muscle cell. We call them satellite cells. I'll introduce more about these cells. So it has long been known that inside this mature muscle cell myofibers, there are lipids. Okay, so these are called little lipid droplets. And then also during regeneration and on a homeostatic condition, there's also fat cells outside this muscle cell. These are adipocytes functioning as a niche supporting cells in the muscle. Now, what I'm going to tell you today is about the lipid droplets in these stem cells, which we don't see in this section here, but we'll see in a second. The reason we're interested in muscle is because, of course, we eat meat, right? All the meat or meats are from a muscle, skeletal muscle, whether it is beef, poultry, or fish steak, tuna, fish, everything is all part of the skeletal muscle, right? The muscle is the meat in agriculture. This is very important, especially like in my department, even though I do biomedical research, but there's a lot of professor here care about how to improve meat production, for example, and the consumers are interested in eating great meat too. Of course, what this group and my research cares is more like health, right? And the skeletal muscle is the key regulator of human health. It occupies almost half of the body mass in our body. And it is crucial for mobility and support. Even you sit in front of your computer, listening to the seminar, it involves muscle, right, that stabilize your posture. And when I sit here and talking to you, I also use my tongue muscle and a lot of muscle, right? So there's this motor and the mobility and the support part makes muscle really important for human health. And then when muscle is not good, it causes disease, such as muscular dystrophy. This is due to degeneration of the skeletal muscle. And what is also known is that in addition to the motor function, muscle also plays a key role in systematic metabolism. So in first, it is the side of the glucose uptake after you eat a meal. About 70% or 2/3 of the sugar that you intake goes into the muscle, and that they are stored as in a glycogen or being used right in the muscle. And the muscle is also a key reservoir of proteins and amino acids. So, for example, I want to say one condition here. In a cancer cachexia patient, the muscle is breaking down to release the protein or amino acids to support the energy demand of the body. So that's one cause of the cachexia, is because muscle stores this protein and amino acids, and when the patient is in a critical condition, they will release these proteins to support the life. And the muscle also is a storage of fat. I talked about the intramyocellular lipids, like in the first cover. This picture here shows better. This again is oil red staining of a cross section of muscle. You can see that there are numerous lipid droplets in the muscle fibers, and this is more prominent in the slow muscle myofibers than the fast fibers here that has a few droplets. Okay. So they relate to the lipid droplets. I'm sure a lot of pathologists that know this phenomenon called the athletic paradox, which refers to that some elite athletes actually has elevated content of lipid droplets in their muscle, while in a diabetic patients, they also have elevated lipid droplets in their muscle. So this is called a paradox, because both a very healthy muscle or very sick diabetic muscle contains a high lipid contents. This may be related to the role of these lipids, how the cell utilizes the lipids. So I'm trying to, hopefully, like the way what I'm presenting today will help you understand a little bit of this. And actually, we have a recent publication, which I don't have time to present today. We actually identify one of this molecule called PRMT5. It's a protein arginine methyltransferase, which transfer a methyl group to a protein to modify protein function. We found that this enzyme, PRMT5, actually is crucial for these lipid droplets turnover in the muscle. So, and identifying this as a molecule that links the lipid metabolism to the contractile function of the muscle. Now, back to the stem cell that we'll be talking about today in the muscle. So the first question is, what is a stem cell? So as many of you know, stem cell is a cell that can divide into more cells, right? And the second, they have the ability to differentiate into mature cell type. In our case here, if a muscle stem cell, they can differentiate into mature muscle cells or myofibers. Or they can also, at the same time, they can also self-renew, going back to a stem cell state. So the stem cell we work on, muscle stem cells, they are called tissue stem cells. Most tissue stem cells are quiescent in a homeostatic state, which means in the absence of injury, these cells are sitting there most time doing not much, right? Nothing. They're waiting for this cue of an injury signal. At that time, this cell will be activated. Once they activate the divide, so we call them mitosis. They divide into two cells, and at this time, these cells often make a decision. Some cells differentiate, so they make the muscle cell that way. They repair the injury in this muscle injury situation. So this can also occur after a few proliferation. That way, there's more cell to differentiate. So this we often call them transient amplification. So other stem cells will immediately withdraw from the cell cycle and they return to a quiescent state. We call this self-renewal. Self-renewal or self-renewal is not a proliferation. Many people are confused. Proliferation meaning the division of the cell. Self-renewal meaning that it is going back to a quiescent stem cell state. I hope this is clear to everyone. So our definition for stem cell, especially for tissue stem cell, is that it is an unspecialized cell that can differentiate into a specialized cell types, and then they can also self-renew to become another stem cell through this cell division process. Okay, the same division can generate differentiating cell and a self-renewing cell. As you think about this situation, you often associate these two different cell fate. We give this a specific name, is the asymmetric cell division. Okay, so when the cell divided asymmetrically, it gives rise to two daughter cells of different fate. This is very, very important for tissue stem cell maintenance. This is less important for embryonic stem cells, where they divide unlimitedly and generate a lot of cell or cancer cells to just keep dividing. But for tissue stem cell, they want to keep the homeostasis balance. Most of the time, the asymmetric cell division is very crucial. So what is controlling this has been a very active research area in the stem cell field. So back to the stellar cells, the stem cell we study in the muscle, we call them muscle stellar cells or MUSC here. I'm going to use this abbreviation. So when you see this, that refers to the muscle stellar cell. So these are stem cell population in the muscle. So originally it was identified in the 1960s. This is the oldest tissue stem cell that has been clearly defined in any stem cell types. Okay, so when the electron micrograph was first invented, they used it by physics to look at the structure of materials. Then biologists begin to use electron microscopy to look at the biological structures. One of the structures they look at is the muscle, right? And of particular interest, they're looking at the muscle of frog. You know, frog, because the frog can jump very well, the muscle is very well studied in physiology. So Alexander Morrow began to use EM to study frog muscle, where he found a cell that's really next to the myofiber. He names stellar cell, okay, because it's almost like peripheral to the myofiber, and he found these stellar cells under the basal laminae. And he also speculated this is an embryonic myogenic cell that is put aside the muscle fiber as a quiescent state, and that this cell is actually important. It may be contributing to the regeneration of the muscle. And this is all proven correct later by scientists. Now we have in our lab, we use, for example, enzyme digestion method. You can take a piece of muscle and put them in collagenase, which will loosen up the muscle bundle. And then you can isolate a single myofiber alive. This fiber is alive. So if you draw attention to this, you can see the myofiber. This, of course, is a very short portion of myofiber. They are striated, right? This is a striated muscle. And then again, you see two colors. One is blue, one is green. That's our standing. The blue is standing for the differentiated cell or myonuclei. And the green is standing for a progenitor cell. In this case, we identify this cell as a stem cell, but this other cell is a differentiated cell. We also have other markers you can use. For example, in this case, we stain the cell with a marker called the PAX7. And again, you can better see this cell and this cell, these two cells are different. You can see this cell is in a compartment with this green, the muscle cell adhesion molecule, and the red is the actual cellular matrix protein integrate. Okay. So you can see that these cells located in the compartment are different from the differentiated myonuclei. I hope you can appreciate, because in this case, you cannot tell the cell are in two different compartments, but this really tells you, and which mimic this situation here. The stem cell is sitting in the peripheral of the myofiber. So this is the myofiber here, okay, isolated myofiber. So if we would cross section a muscle, this one shows a little bit better in the muscle. And now we have a different marker. We marked the cell in GFP, green fluorescent protein, and also label the cell with a marker, the red marker here. You can see that these cells are under this white line, which would be the basal lamina here. Yeah. So it's under the basal lamina, but outside the myofiber. It's not part of the myofiber. As you can see, the fluorescence is different, right? I hope all this convinces you this is an independent cell population that we study, and then we know they are stem cells. And again, under the EM cross-section, you can see the cell is separated from this cell. Is this a gap in between this cell and the myofiber? And also the nucleus shape is different. Cellular cell nucleus is smaller than this. And then you can also appreciate the difference in size here, okay, and in here, the difference in the size. So it is no stem cells. These stem cells are a little bit smaller than differentiated cells. And then the chromatin is much darker under EM. So this is indicative of inactive transcription. As I said earlier, in the quiescent stem cell, they're mostly sitting there doing nothing. These cells inactive, and so the heterochromatin usually indicates an inactive status. So this is, as I said, this is a stem cell population. It has been widely exploited in our field to target the stem cell either by transplantation, cell transplantation, or by targeting the signaling transduction of these cells to ameliorate muscle regeneration. So this is a review cover that I wrote a number of years ago. So it has been a very active research area, how we can utilize our knowledge about this stem cell population to benefit muscle regeneration of disease patients like muscular dystrophy patients. So now I'm going to talk a little bit how we study these stem cells. Okay, one way we study these stem cells, which is also unique in our field, is that we can isolate a single myofiber. I mentioned that earlier, you can digest a whole bundle of muscle with a collagenase, and then you can isolate a single myofiber. Again, this is a picture of myofiber, a portion of a myofiber. Okay, myofiber can be very long. Some muscle in your body has myofiber as long as 1 meter. Okay, so others are mostly shorter. But in mice, we can take muscle myofiber as a few centimeters long to study in culture. So this fiber is still alive. You can grow them in culture because the fiber carries this stem cell on it. Remember, I showed in the previous slide, the stem cells on the myofiber, although they're too small to see here. But if you look at very closely, like if you magnify any of the region, you can see stem cell on the fiber. In this case, we don't see the background fiber, but we only see the stem cells. When we grow this in a culture condition, in the first 24 hours, the cell are not divided. You only see one cell, okay? So we have reliable markers so we can label the cell. One of the marker here, I'm going to talk about the marker now, is PAX7, pair box protein. This is a transcription factor in the nucleus, okay? And then another marker is MyoD. Some of you may know MyoD. Myo means muscle, D is ABCD, right? So this is the first factor identified that is a muscle stem cell, myoblast specific. And then you can transfect a fibroblast with MyoD to make them into a muscle cell lineage. So this really established the platform for induced pluripotent cell now. So all the pluripotent cell concept was to force them to express a gene to change one cell to another. But this experiment that was done in the 1980s, that's like 25 years before the iPS cell. But it gives a concept. You can express a single transcription factor to make a one cell type into another cell type. And this is the first study in the literature showing cell fate can be reprogrammed by a transcription factor, which is MyoD here, okay? So we use these both markers to mark stem cells. And as you can see, if they have both marker green and red, they appear yellow, right? So then this cell would divide in less than 2 days, 48 hours, they become two cells. I hope you appreciate here that even in the two cell state, these two cells, if you look at the shade of green and red, which is the relatively yellowness, you can see there was a slight difference. This is a little bit greener than this, right? And then you can see more here. This is much greener than that. This is still two cell stage. And this, I want to say this because this was suggesting that there was some kind of signaling going on in between the cells that makes these two cells different, right? One red cell and one green cell. And this is a typical sign of asymmetric cell fate, right? And cell fate is segregation. So we're talking about the cell fate. The question is what makes this cell a red cell, which we actually call this a self-renewing stem cell. What makes this cell a green cell, which we call them the myogenic myoblast. Then we differentiate into muscle. You can see here when they have 6 cells, it looks much clearer. There was a green cell and a red cell. And this is for 3 days, 72 hours. This cell has already formed a big cluster of cells. And then some cells are red, some are green, and then there's still yellow cells. And this represented a very, very good model for us to understand the stem cell fate, because you can grow fiber and waiting for this stem cell to divide, and then ask what is the fate, right? Based on these two markers, red or green. And then we can associate genes or signal to this different cell fate to understand what controls to this different cell fate and segregation. So I just want to know that I cannot see your questions because I'm talking. But if you have questions, please feel free to just ask, right? Turn on your microphone and ask, because otherwise I cannot see. Or you can wait until the end of my talk. So this is a culture model. The need of this model is that this stem cell is still on the host myofiber. So it is like, even though it is a culture condition, but it's actually mimicking the in vivo situation, right? It's almost like the same, the muscle is inside the body, right? Of course, it is not the same because it's in culture. but it has partially the same. So we think this is a very unique and a nice model, almost like an organoid people now use in culture. Many people create. Another way we study the muscle stem cell is in vivo, where we can inject a snake toxin like a cardiotoxin into the muscle. Or you can even inject a compound like barium chloride. This is compound that causes depolarization of the muscle, which causes hypocontract and the muscle cell death. And because the death of the muscle cell will trigger those stem cells to divide and repair, right? I mentioned that they are stem cells. So this is what the muscle look like before you inject the toxin. And then right after the toxin injection, this is 0.5 days after post-injury. DPI is day post-injury. So you can see the muscle begin to degenerate. So at 2 days, much degenerated. You can see all this white space here. At that time, these stem cells, even though we cannot tell which one is a stem cell, but one of those, some of those blue marked cell nuclei, they actually start to proliferate. You can see there's so many more nuclei here than in here, right? This stem cell, many of these are stem cells, begin to proliferate and regenerate the muscle. You see, after 21 days, the muscle is largely repaired compared to the pre-injury level, right? The only difference is now you can still see nuclei in the middle of the myofiber, like I showed in the beginning. In the non-injured muscle, those nuclei are on the peripheral, but this regenerating muscle has central nucleation. Okay, so this also pathologically, this also gives us a mark or indicator of muscle regeneration, because we can easily tell which muscles regenerate compared to a non-injured muscle, even though the overall appearance seems to be similar. But this nuclei, they are stuck in the middle. They never goes to the peripheral, which also suggests that there was a difference between regeneration and the development. Developmentally, the fiber can go to the peripheral, but during regeneration, it seems to be stuck in the middle. So this in vitro culture model and the regeneration model is what we commonly use to study stem cell mediated regeneration in muscle. Okay. So I want to make you appreciate this here. So I want to go back a little bit. This is isolated from a stage like a 5-day, when you see a lot of this very small regenerative muscle with central nuclei. So what we did is we isolated single myofiber. Of course, this is all cross sections. We isolate single myofiber from this stage and then look at the cell contribution to the regeneration. On the top is a non-injured fiber, a fiber from a non-injured muscle. You can see on this length here, you see one solid cell, which is green, right? That's clear, non-injured. So this green is marked by a fluorescent protein, yellow fluorescent protein, and all this blue are myonuclei, okay? Now you look at the fiber at the 5-day. Actually, this one says 4-day, is a day before the 5-day. At this time, you can see there's a chain of central nucleation, right? Central nuclei, as I told you repeatedly earlier, this is a very well indication of regenerating muscle. You can actually see the injury here. But what you can tell, the difference is there's so many more of this GFP marked solid cell. So these are the stem cells, they are actually dividing, and you can see these branches, they also moves around to repair injury in the muscle, right? Isn't that a very nice system, right, to study? What is even better, if we look into these cells, you can see some of these cells are dividing. They have 2 nuclei in them. These cells already, maybe these are also newly divided, but these are still kind of almost not completely segregated. So this is a really nice system to allow us to understand the cell fate segregation, because in this system, what I can see is there were cells that express having this GFP. In this case, the YFP is an indicator of MIP5, and the the lack of YP means they don't have the MYF5 marker. So you can see there's a cellular heterogeneity. Some cells is green, some cells does not, right? So if we look at these dividing cells, we can see cells with green, cells without the green. Then we can potentially study their asymmetric cell fate segregation, right? Or the other question to ask is that this green cell and the non-green cell, are they from the same cell division, right? In here, we are not sure. But what was nice is that when I was a postdoc fellow, I made the discovery that this cell actually can indeed divide asymmetrically to give green cell and red cells, which we call self-renewing the commitment cell. So in here, as you can appreciate here, there's a central nuclear indicating there's regenerative muscle. Now you can see these two cells here, right? They were in the same compartment here. So all surrounded by the basal laminin and the green cell adhesion molecule in the same compartment. So does these two cells here. And then now what you can see is now you have red cell and green cell here, okay? I'm just simplifying red, PAC7, green, MYF5, but we just use green and red to show these are two different cell fate, okay? So there is cell dividing asymmetrically in vivo. This is isolated in the regenerative muscle in vivo, not cultured, okay? So this is really the first time demonstrating a muscle stem cell can divide asymmetrically, give a self-renewing cell and the commitment cell, okay? That's why this paper is published in Cell. So just to summarize this part of the result, is more like a background that we published many years ago. But this result is highlighted in the developmental biology textbook. So what we shown is that the cell can divide symmetrically, give rise to two cells of similar fate. They are either both red or both green, right? They can also divide asymmetrically, give rise to two cells of different cell fate, a red cell and a green cell. And what we establish is that this occurrence of the asymmetric cell division is mostly occurring in the apical basal relative to the myofiber. So which suggested that there was something in this compartment that determining the cell fate. Specifically, when the cells are apical basally localized, the two cells are in a different position relative to the myofiber and to the extracellular matrix. Because these cells are close to the myofiber, these cells are away. But when in parallel, both cells are contacting the myofiber, and they are mostly similar cell fate. They're either both red or both green. Okay, so this establishes really a niche. Niche is this microenvironment that could have impact on the cell fate when the cells are different niche and two different kind of outside or inside. They have different cell fate where the cells are both in parallel to the myofiber. There's no such thing called outside or inside. They have the same cell fate. I hope this is clear to everyone. So we are trying to establish this. So I think so in this niche here, what we don't see is the outside. There's a lot of stuff outside. If you look at a muscle and these stem cells, in the outside of this, like extracellular matrix, there's a lot of microcapillary, right? Blood vessels and the lymphocytic vessels here in the muscle. So there's a lot of structure we are not seeing. This is too simplified. But the idea is that when they're exposed to these vessels on the outside or exposed to the host myofiber, they are facing two different microenvironments, and this could be a reason for the cell fate segregation. Okay, this is something from outside. There's also stuff, this signal from outside triggers signaling inside the cell that causes the different cell fate, right? So one of the active research area in our lab is to understand what is inside the cell that's causing these two cells to take different cell fate. This is a key question in stem cell biology, as I mentioned. So the rest of the study, I'm going to talk about the lipid droplets because I think that this is quite exciting here. So what it shows, what I want to show you is that these are tiny droplets of lipids in the cell are mostly found in fat cells. Like in the fat cell, you can see very large droplet. They're made by the ER, and we are talking about droplets in the solid cells are much smaller. Like here, these of course these are not all solid cells, but you can see appreciate the tiny droplets. That's what we are looking at today. It starts with the observation my postdoc fellow made is that when you put this cell on the myofiber, when they actively divide, right? We showed that process. What he found is that lipid droplet emerge in these cells during this process, as you can see here, right? So what you can see better is that you can use confocal imaging. You can really see the droplets in them at different time points when the cell before or after differentiation. And this is a real presence of droplet because we can mark with the membrane lipid droplet protein perilipin 1. You can show these lipid droplets in them. And we can also use the non-invasive Raman imaging technique here to co-localize these body P droplets with the Raman signal. So this is all showing that we are actually validating their droplets in this muscle cell. And this is another way, based on the lipid profile, you can see this droplets has a light emission like profile, like we call spectrum profiles that's identical to lipid droplets in this newly divided stem cells here, okay, on the myofiber. So this is all culture. Does that occur in vivo during regeneration? So I talk about the regeneration day 0, 7, and 30 when regeneration finished, right? This timeline is already presented before. But you can see that the lipid droplets are mostly bounded at day 7 during the regeneration. We can even use a FACS to sort cells based on the lipid droplet content. So the lipid droplet high versus low. And then we can also look at the cell under the electron microscope and see the actual lipid droplets in these solid cells during the regeneration, but not before or after, okay? So we're showing here is that lipid droplets are highly dynamic in the in vitro and in vivo. So what might be the function of these lipid droplets, right? So we talk about how the different cell orientation is related to cell fate. So what interestingly we found is that lipid droplets can also be asymmetrically segregated in cells when they exhibit different cell fate here. Right, this is quite exciting. This has never been reported before, that when the cell divided into 2 cells, one cell has more droplet than the other cells. So that's quite exciting. And then we use the same protocol here. When you culture a cell cluster, you have a red and a green cell. We can also find that the red cell has fewer droplets than this green cell, which is a blue cell here. So the droplets are mostly with the blue cell. So this part suggests that the lipid droplet distribution is associated with cell fate. So I'm going a little bit faster because these are all published. I was slow in introducing our system, so everyone was on the page, right? So the next question was, can we perturb droplets to affect the stem cell fate? So we used the 2 different methods. One is to inhibit the biogenesis of droplets. One is to inhibit the degradation, okay? First, we use a compound to block the lipid synthesis, which will actually reduce the droplet in the cell here. Droplet is reduced. But accompanying this, you can see the cell number is reduced, but most prominently we see an increase in the self-renewing, which is the red cell, the ratio, not the absolute number. So this is kind of a percentage, total is 100%. The ratio of this self-renewing cell is increased when we block the lipid droplets. So which means that promotes the quiescence, okay? When we block the degradation by knocking out an enzyme that's key, crucial for the degradation, we found that the cell the lipid droplet content is increased. At the same time, there's more cell differentiating, which is the green cell here. Okay, so it will affect the self-renewal, reducing the self-renewal, and this can also cause a defect in the muscle regeneration. Here, I'm just going fast. So when we block the droplets degradation, the regeneration. So this is some pathology. You can see that there's difference. This time point is similar to this. This is defective regeneration in that case. So when we perturb droplets, it in fact affects the cell fate and the regenerative function. The next question we did is we actually directly isolated lipid droplet low and high cells and then asked whether they have different function, right? So I mentioned earlier, we can separate the cells based on the lipid content. So this is our strategy. We separate the cell. Now, the next thing we did was to inject this cell into a regenerative muscle to see whether their contribution to muscle regeneration is different. So this is the outcome. So because all this transplanted cell is labeled in red, more red, meaning that they make a better contribution to the regeneration. You can see that on the top is the lipid droplet low, and comparing to the lipid droplet high cell. The low cell makes more red cells, and it also makes more solid cells. This part is showing that this lipid droplet low cells actually regenerate the muscle better than the lipid droplet high cells, okay? So they do have different regenerative potential here. So this is very cool. So I think, so we have about 10 minutes. I'm going to talk a little bit about the unpublished result. What might be this lipid droplet do in this cell to affect the regeneration? So lipid droplets are a storage of fatty acids in the circulation. It goes into the cell, store lipid droplets. When there's a need, they can release fatty acid, and then they engage in a process called fatty acid oxidation in the mitochondria. So this is what you see, fatty acid oxidation here. So fatty acids first has to be transported inside the mitochondria through this enzyme CPD1 and CHCT. Once in the mitochondria, it is going through a process called beta oxidation, okay? And then generate the acetyl-CoA used in the TCA cycle to generate energy. So these are the three key enzymes for FAO. And because there are different isoforms, I think that we start to target the CPD2 here, okay? So the question was that if we block this, does that affect the cell function? So just remind you, the lipid droplets increases during cell differentiation. And then we can see the CPD2 expression also increase during this same timeline, okay? And you can see this is a transcript, that's the protein level of CPD2 both increase along this, suggesting there's a good correlation between the CPD2 and the lipid droplets. So we developed a mouse model to specifically knock out the CPD2 in the cellular cells. And this is the design. We injected tamoxifen here to knock out this in the cellular cells only. Then we injure the muscle to look at regeneration. You can see that pathologically, this is actually worse than that. I don't have too much time, but just trust me, this is delayed. We have been looking at this a lot of times, this defect in regeneration. What happened to the FAO? So this is a labeled C13 labeled fatty acid pulsing into the cell to see how this fatty acid is incorporated into the TCA cycle here, okay? So, but this is a knockout, CPD2 knockout, this is wild type. You can see knockout has a reduced incorporation of this fatty acid into the TCA cycle, showing there's a defect in the TCA cycle. And this is much more clear with the seahorse analysis, where you add different compounds, looking at oxygen consumption. When they consume more oxygen, meaning that they have more active metabolism. You can see the blue trace is a knockout. They have consistent and lower oxygen consumption, meaning that lower metabolic rate in these cells. what we also found is that because the TCA cycle generated acetyl-CoA, right? We talked about acetyl-CoA here. The acetyl-CoA level is very low in here, and the acetyl-CoA is a donor for acetylation of proteins. We look at acetylation in the knockout is actually reduced. So we actually specifically the acetylation of PAC7 here is reduced. So we kind of ask them how does the change in acetylation change the cell function. Just a long story short, this is an RNA sequencing study where we looked at the CP2 knockout versus wild type SADA cells, looking at what is the difference in the gene expression. But we also correlated this difference to a set of data showing how PAC7 control, like PAC7 targeted gene. This merge is generated 90 genes that we think is PAC7 targeted genes that's affected by CPT2 knockout. Among this 90 was showing here, what you can see is that normally genes activated by PAC7 in wild type, yellow meaning activate here, is reduced. Whereas gene that's repressed by PAC7, in wild type is elevated in the knockout. This is some examples of how this gene change, which means that this knockout deregulates PAC7 targeted gene, which means because PAC7 itself regulates gene expression, means that it actually affected the PAC7 function, okay, the knockout of CPT2. And then we can actually add acetate, because acetyl-CoA is acetyl donor, right? We can add acetate into the culture and the condition to kind of rescue the respiration and the SADA cell number here. This is too complicated. I'm going to skip that slide here. Just to bring to the summary here. What we show in this part is that FLAO, or fatty acid oxidation, potentially of lipid droplets, right? Because the fatty acid is from droplets. It's very important for SADA cell energy, metabolism, and signaling. Specifically, if we knock out the CPT2, we are blocking the FLAO here. What this consequence is that on one hand, it actually reduces acetyl-CoA, and reduction of acetyl-CoA will impair the TCA cycle. This is a reduced production of ATP, but it is also increased and reduce the acetyl-CoA, also reduce acetylation of this key gene that's important for SADA stem cell function. And this affects SADA cell regeneration. Okay. So this actually really links the lipid dynamics to the energy metabolism and gene regulation, right? So we propose this model here that in the quiescent SADA cell that do not have lipid droplets, when they activate, they start generating droplets. When they divide, the generated cell with high or low droplets. The low droplet cell are self-renewing stem cells, the higher cells differentiate. If you block this lipid droplet from formation, you kind of have a relatively more self-renewing cell. If you block in the catabolism, it causes more droplets. That time you are depleting the self-renewing cells. At the same time, it causes the oxidative stress and all kind of condition here that's affecting SADA cell regeneration. Okay. So the overall consensus is that when we reduce the lipid droplet, it promotes self-renewal. But if you increase, it stimulates the activation and proliferation. So we are very excited to actually find out when we publish the paper, there's another group studying neural stem cell also found that by reducing lipid droplets in neural stem cells, they also promote quiescence, and increasing the lipid droplets stimulate proliferation. There's other cells, similar study in cancer stem cell, in intestinal stem cells, cancer stem cell. Lipid droplets plays a similar role in the self-renewal and the differentiation. So these are some studies I'm going to skip, just showing similar function of lipid droplets in stem cells of other types, including embryonic stem cells, and also in cancer cells. So in the final slide, I want to just to sum up that stem cells can divide symmetrically to give rise to cells of the same fate, or the divide asymmetrically to give rise to a stem cell and a committed cell, a differentiating cell. And in the literature, this was supposed to be determined by this so-called cell fate determinants, which are the white dots here. When these cell fate determinants are equally segregated to generate a cell fate, when these determinants are unequally inherited by the two cells, then they generate a different cell fate. So in the past, in the literature, this cell fate determinants has been supposed to be proteins, for example, PAS3, mitochondria, DNA, or centrosome. Now, what we are showing for the first time is that lipid droplets can also be one of these cell fate determinants during cell division. So I hope this is a clear cell message for you, and we are actively working on this, trying to understand better how this determines cell fate. So this is my group at the Purdue, and the work is mostly done by, actually I don't have these two pictures here, but mostly done by Candace and a postdoc who is not here from, he's currently an assistant professor in Florida. So I also want to acknowledge all the funding sources for my research. With this, I'll be happy to entertain any questions in the next 10 minutes or so.
Thank you, Shihuan. This is such an interesting talk. Yeah, so we're now open to questions.
I'm going to try to stop the share so I can basically see some questions. I don't know if there's any chat questions.
Let's see.
And you can also type question in the chat box. So ask. Just turn on your microphone and ask.
Okay, so while we're waiting for the questions, I actually you just mentioned that this lipid possible link lipid. Some people also propose the lipid metabolism and the neuron stem cell, the neuron analysis. So as you know, the only risk factor for sporadic Alzheimer's disease is ApoE4, which is a lipid transport protein. And I think there is, although it's still controversial, but I think there's now evidence, at least in the animal study, that shows in Alzheimer patients that actually the neurogenesis is reduced. And there is actually some lipid accumulation in the subventricular zone of ependymal cells, which are also similar to the adipose tissues. Maybe also a niche for the stem cell neurogenesis. So there's also evidence to show there's actually lipid accumulation in those glial cells. So I just want to get your insights, whether you think this is real, there's at least a possible association with the neurogenesis.
Yeah, that's actually a great question. I think I'm glad that you mentioned this. This is completely actually possible. Like what there was the neural stem cell paper was published a month before ours in Nature Communication, clearly showing the link, or at least the function of lipid droplet in the neural stem cell. Of course, in the Parkinson's disease, we are talking about mature neurons. And the ApoE you mentioned, involved in transfer of this cholesterol, right? Lipids. And the lipid droplets also contains, other than TAG or triacylglycerols, it also has cholesterol and other lipids, ceramides. And understanding the link between lipid droplets and Parkinson's disease and neurological defects, I think that would be quite exciting. I'd be happy to contribute, or if you need any reagent to study lipid droplets in your
that'd be great.
model. Yeah, in neurological diseases.
Yeah, this is definitely something, yeah, we're interested to work on.
Questions?
Because other questions.
I think sorry, we can't hear you. Jess, I think you're muted.
Can you hear me now?
Yes, yes.
Yes.
Sorry about that. So I saw that you used the Raman imaging to kind of differentiate the lipid, and it looked like there were some like hot spots in the lipid body. Are you able to differentiate, or do you see like different patterns within the lipid bodies with the different lipids, cholesterol and trigs of that nature?
Yeah, that's a great question we are actively pursuing. So now that you ask this, I want to expand this. So it's funny that people has shown in yeast and yeast model, even lipid droplets in the same cell could have different pattern in their protein expression and the lipid content. And that Raman imaging would be really great for us to understand the heterogeneity. Because when we look at the lipid droplet segregation into two cells, we are looking at the quantitative. There could also be qualitative difference between them. And that's really great question. I thank you for mentioning this. We don't know yet for now. You asked a good question. I don't know the answer, but we are actively looking at. Yeah.
It looks like you're on the right path to solve it, though. So
yeah, we have a 5-year NIH grant looking at this now. So after, obviously, the reviewers are also excited about this project. Yeah.
Thank you.
Thank you for the question.
Okay, so if no more questions, I want to thank you again, for this really excellent talk and really sharing with us your exciting findings. And we hope we can in the future possibly work together. Thank you so much.
Yeah, thank you for the opportunity to introduce our work.
Sure.
Goodbye, everyone.